Have you ever wondered how something as complex and beautiful as life first flickered into existence on this ancient planet? Today, we journey into the heart of that mystery, into the very beginning of beginnings. If this story moves you, like, and share it to keep the questions alive in others. Let us now return to a world where Earth was young, silent, and waiting. In the earliest epochs of Earth's formation, the stage was anything but welcoming. The planet was a roiling sphere of molten rock and churning seas, battered by meteor storms and seared by ultraviolet radiation. Yet in this chaos, beneath the violence of tectonic fury and a sun still young in its fusion fire, the first ingredients of life began to gather like whispers before a symphony. The oceans, dark and warm, stretched endlessly beneath a hazy sky, holding within them a restless chemistry. The crust of the earth cooled in fits and starts, birthing volcanic islands and deep oceanic trenches where hydrothermal vents hissed and steamed, exhaling mineral-rich breath into the watery darkness. Life, as we know it, requires structure, information, and energy. Somehow, against astronomical odds, these elements danced into harmony. It is in this improbable ballet that science searches for the origin of life, the moment non-living matter was transformed into living substance. One of the most prominent theories that attempts to explain this transition is known as abiogenesis. This hypothesis holds that life arose naturally from simple organic compounds, without the intervention of pre-existing organisms. It is not a question of spontaneous generation, as once imagined by ancient thinkers, but a gradual unfolding, a layering of complexity over unimaginable spans of time. The Miller-Urey experiment of 1952 offered the first glimmers of empirical support for this possibility. Stanley Miller and Harold Urey simulated what they believed to be the conditions of early Earth, an atmosphere rich in methane, ammonia, hydrogen, and water vapor, all subjected to electric sparks that mimicked lightning. Within a week, their closed system began to yield amino acids, the very building blocks of proteins, which themselves are the machinery of life. It was a revelation that life's components might arise from inorganic chemistry under the right conditions, with energy as the catalyst. Yet amino acids alone do not make life. The mystery deepens when we consider how these small molecules could have organized into complex macromolecules, proteins, nucleic acids, membranes, that possess the capacity to replicate, mutate, and evolve. Central to this enigma is the question of information. How did nature inscribe the code of life? The answer may lie in the RNA world hypothesis, a theory that posits ribonucleic acid, or RNA, as both the carrier of genetic information and the catalyst for chemical reactions in early life forms. RNA has the unique capacity to act as both a template for inheritance and as a ribozyme, a molecule capable of accelerating reactions. This dual functionality suggests that RNA may have been the molecule that first bridged the gap between chemistry and biology. In deep time, it is imagined that self-replicating strands of RNA formed within vesicles of fatty acids, floating in Earth's primordial soup. These protocells, as they are called, may have been the earliest lifelike entities, capable of growth, replication, and even rudimentary evolution. But where did these fatty acids, sugars, and nitrogenous bases, the ingredients of RNA, come from? Some scientists propose that they arose in Earth's early environment through naturally occurring chemical pathways, driven by the heat of hydrothermal vents or the energy of ultraviolet radiation. Others suggest a more cosmic origin. The panspermia hypothesis entertains the notion that organic compounds or even primitive microbes may have arrived from space, delivered to Earth by meteorites or comets, which have been shown to carry amino acids and other complex molecules. If true, this would shift the birthplace of life beyond our planet, embedding it in the story of the stars. One famous meteorite, the Murchison meteorite that fell in Australia in 1969, contains more than 70 different amino acids, some of which are rare or non-existent on Earth. The implications are staggering. If the seeds of life rained from the heavens, then perhaps the universe is not silent but fertile. Even so, the leap from molecule to metabolism remains among the greatest chasms in our understanding. Life is not merely a collection of chemicals, but a system that maintains itself through cycles of energy, exchange, and repair. Some researchers believe that life may have emerged near deep-sea hydrothermal vents, where mineral-laden water seeps through porous rocks, creating natural compartments. These microenvironments could concentrate molecules, catalyze reactions, and provide the gradients of heat and pH necessary for life's machinery to begin. These structures, known as alkaline vents, exhibit a kind of natural order that mimics biological cells, compartments within compartments, capable of directing chemical flows. Another theory suggests that life began on the surface, in shallow pools or tide zones that underwent cycles of drying and rehydration. In such settings, simple molecules could be concentrated by evaporation, bonded into longer chains by the heat of the sun, and fragmented again by sudden rains, an iterative process that may have driven molecular evolution. Life, in this view, is not a sudden event, but an emergent property, something that happens when matter is arranged just so, under the right pressures, and given enough time. Through all these competing theories, one theme echoes persistently. Life is the outcome of order rising from disorder, of complexity born from chaos. Whether in the abyss of the oceans or the blast of meteorite impacts, life found a foothold, a starting point. And from that fragile beginning, somewhere between chemistry and biology, the great narrative of evolution unfolded. Earth, at the time, bore no forests, no insects, no vertebrates. There was no oxygen in the air, no blue in the sky. The surface was scorched, the seas clouded with minerals, the crust fractured and restless. And yet, in some concealed corner of this tumultuous world, the spark was struck. What followed was not instantaneous brilliance, but a slow bloom, a crawling, silent revolution that would, over eons, build cells, organisms, ecosystems, and thought. As the world turned under a dim sun and clouds of volcanic breath, time moved. Like a slow tide, indifferent to the fragile beginnings hidden in the shallows and the depths, what had once been inert now hinted at purpose. Molecules that had danced randomly across the prebiotic seas began to align with subtle tendencies, building scaffolds of logic within the chaos. From the first whispers of self-organization, the architecture of life began to take shape, not in leaps, but in microscopic steps shaped by the forces of physics and chemistry, written in the quiet grammar of nature. The next profound chapter in this emergence was compartmentalization. Life, in its earliest forms, could not remain diffused and unbounded. To persist, it needed containment, walls to preserve its internal chemistry from the vagaries of the external world. Here, the concept of the protocell becomes central. These primitive enclosures, made from simple lipid molecules, formed spontaneously in water, assembling into bilayer spheres. Such vesicles mimic cell membranes, acting as selective gates between an inner world and the outer environment. Inside these protective shells, the fragile molecules of early metabolism and replication could begin to co-evolve. The environment within a protocell stabilized chemical reactions, encouraged concentration, and nurtured the seeds of interdependence. As strands of RNA began to replicate, mutate, and interact with proteins, a new kind of chemistry took root, one no longer shaped solely by the external world, but increasingly governed by the internal logic of the protocell itself. Life was gaining momentum, and with it complexity. With replication came inheritance, and with inheritance the possibility of evolution. The great engine of natural selection, operating on molecules instead of animals, began to churn slowly. Not all protocells were equal. Some replicated faster, some resisted environmental disruption better, and some evolved internal reactions that harvested energy more efficiently. Gradually, through competition and accident, primitive metabolisms began to arise. Metabolism, life's heartbeat, is the process by which organisms convert energy into usable forms. In today's world, this involves complex networks of enzymes, proteins, and cofactors. But in early Earth, metabolic pathways likely began in simpler fashion. One hypothesis proposes that iron and sulfur minerals at the bottom of ancient oceans catalyzed reactions between carbon dioxide and hydrogen, producing organic molecules and energy gradients. These geochemical processes may have served as the prototypes of the citric acid cycle, glycolysis, and other essential biochemical loops. The idea of surface metabolism, where mineral surfaces guided and catalyzed early reactions, suggests that life's first breath was not a cell, but a network, a field of reactions flowing over a surface, tethered to the rocks and sustained by gradients in heat, pH, and electron potential. These metabolic systems, once encased by lipid membranes, would have taken their first steps toward autonomy. Energy, matter, and information began to coalesce into systems capable of growth, change, and persistence. Parallel to this metabolic, blossoming genetic material was evolving. RNA, the early master of both information and catalysis, was not perfect. It was fragile, prone to error, and inefficient compared to the molecules that would follow. Over time, DNA, a more stable molecule for storing genetic information, began to emerge. The transition from RNA-based life to DNA protein life was not abrupt, but gradual and overlapping. Ribosomes, the complex machines that read RNA and build proteins, are fossils of this transition, retaining ancient RNA components in their very cores. Proteins, composed of amino acids, brought new power to life's toolkit. They could fold into intricate three-dimensional shapes, performing specific tasks with precision, cutting molecules, building structures, transporting substances. The evolution of the genetic code, the set of rules by which RNA sequences are translated into amino acids, marked a monumental leap. From a world of chemistry emerged a language of life, capable of expressing nearly infinite forms. As protocells grew more sophisticated, they began to interact, compete, and perhaps even cooperate. Horizontal gene transfer, where genetic material moves between unrelated entities, likely played a vital role in this era, allowing the sharing of successful innovations across different lineages. The landscape of early life may have resembled a tangled web more than a tree, with evolutionary lines crisscrossing, merging, and diverging in unpredictable ways. Eventually, a remarkable event occurred: the rise of the last universal common ancestor, or LUCA. This was not the first life form, nor the most complex, but it was the one whose descendants would populate every branch of the evolutionary tree. LUCA was likely a simple single-celled organism, possessing DNA, RNA, ribosomes, proteins, and a lipid membrane. It had a basic metabolism, used ATP for energy, and existed in a world already rich with molecular diversity. The search for LUCA is an ongoing endeavor, pursued through comparative genomics and molecular archaeology. By analyzing genes common to all known life forms, scientists have reconstructed a minimal blueprint of LUCA's machinery. This ancestor probably lived in a hydrothermal environment, perhaps near alkaline vents or in warm subsurface waters. It thrived in an anoxic world before the rise of oxygenic photosynthesis, and it likely relied on simple molecules like hydrogen, carbon dioxide, and sulfur for its energy. From LUCA, the great diversification of life began. Some descendants became bacteria, mastering fermentation and simple respiration. Others evolved into archaea, capable of surviving in extreme environments. Still others laid the groundwork for the emergence of eukaryotes, the complex nucleus-bearing cells that would one day give rise to plants, fungi, and animals. Each of these branches would trace its lineage back through a common molecular ancestry, to a time when life was not yet divided by species or kingdoms, but united in its rudimentary struggle for persistence. In this ancient world, evolution was a quiet sculptor, etching the patterns of future biodiversity into the microcosms of early Earth. And so, the Earth turned, carrying within it the echo of a transformation that would reshape the surface of the planet. Life, once a flicker in the chemistry of oceans, had become a presence still microscopic, still vulnerable, but persistent. It clung to rocks, bubbled in the warmth of vents, drifted in currents, and divided silently beneath the gaze of stars. The great silence that once filled the Earth had been broken, not by noise, but by replication. In that first moment when a cell copied itself and passed along its pattern, the history of life truly began. And with it, the slow, majestic unfolding of complexity that would lead to forests, whales, beetles, and minds capable of asking how it all began. Life, in its infancy, was a symphony of invisible rhythms, replication, mutation, competition, carried out in miniature by cells no larger than dust. Yet within these tiny chambers, universes of potential stirred. Time, measured not in days or years but in the slow spirals of genetic drift and adaptation, allowed this fragile phenomenon to endure and evolve. The world outside remained harsh, bombarded by ultraviolet radiation, scorched by volcanic breath, and stirred by turbulent tides. But the world within, the microcosm of the living cell, had begun to master its own alchemy. In this stage of evolution, the lineages of life were still simple. Bacteria and archaea dominated the scene, organisms without nuclei, without organelles, but with astonishing biochemical versatility. They thrived in conditions modern life would find inhospitable. Acidic pools, salt-saturated lagoons, frozen wastes, and boiling undersea vents. Their resilience was not just a feat of biology, it was a testament to the adaptability encoded in life's molecular grammar. Among these early lineages, the division between bacteria and archaea slowly became apparent. Though both lacked nuclei, their membranes, genetic transcription machinery, and metabolic strategies began to differ. Archaea developed strategies to stabilize their structures in extreme environments, while bacteria diversified their metabolic abilities, colonizing every conceivable niche. This divergence was quiet, but significant. It laid the foundation for a new evolutionary experiment that would one day give rise to the complex eukaryotic cell. Photosynthesis emerged as one of the most transformative innovations in the history of life. Some bacteria learned to capture sunlight and use it to split water molecules, releasing oxygen as a byproduct. This process, oxygenic photosynthesis, began with cyanobacteria, tiny architects of planetary change. As they multiplied across Earth's shallow waters, they flooded the atmosphere with oxygen, initiating a transformation that would ripple across billions of years. The rise of oxygen marked a turning point known as the Great Oxidation Event. This was no gentle shift. It was a planetary upheaval. Oxygen, toxic to many of Earth's anaerobic inhabitants, caused mass extinctions. But it also unlocked new possibilities. Oxygen made aerobic respiration possible, allowing cells to extract far more energy from organic molecules than fermentation alone could provide. It was a biochemical revolution, one that enabled the rise of larger, more complex life. Still, complexity in its modern form required something more, a fundamental restructuring of the cell. Somewhere in Earth's primeval oceans, a dramatic event unfolded. An archaeal host engulfed a bacterial guest. Instead of digesting it, the host formed a partnership with its captive. This was endosymbiosis, a mutualistic arrangement that gave birth to the first eukaryotic cell. The engulfed bacterium became a mitochondrion, an energy-producing organelle that fueled the eukaryotic cell's metabolic needs. This moment was a hinge in life's narrative. With mitochondria generating energy more efficiently, eukaryotic cells could afford to grow larger, evolve internal compartments, and develop complex regulation systems. Nuclei formed to house DNA, endoplasmic reticula emerged to process proteins, and Golgi bodies packaged them for use. Life was no longer a simple bag of chemicals. It had become a structured city, a vast architecture of function and purpose. These eukaryotic pioneers diversified slowly. Some remained single-celled, but others began to aggregate. Colonial organisms formed, loose groups of cells living together, sharing tasks but still independently viable. Over time, in some lineages, this cooperation deepened. Cells began to specialize and surrender autonomy in favor of unity. Thus began multicellularity, one of evolution's most profound transitions. Multicellular life was not born of necessity, but of opportunity. With oxygen abundant and energy efficient, with internal complexity already developed, the stage was set for organisms to grow, adapt, and diversify as coordinated wholes. Specialized cells allowed for division of labor. Some focused on movement, others on digestion, reproduction, or defense. Tissues formed, then organs. Nervous systems arose to sense the environment, muscles to move, and digestive systems to fuel the whole. In the depths of the oceans, strange new forms emerged, soft-bodied creatures gliding, burrowing, feeding. The Ediacaran biota, the first large multicellular organisms, painted the seafloor with leaf-like fronds and quilted geometries. They lacked bones, eyes, or true limbs, but they hinted at the shapes that evolution would soon explore with greater intensity. Then, in a blink of geological time, the Cambrian explosion shattered the stillness. Over a few tens of millions of years, nature experimented with forms in a fevered burst of creativity. Trilobites with armored shells, segmented worms with bristles, jawless fish with notochords, all sprang into existence. Nearly every major animal phylum appeared in this evolutionary tempest, populating oceans with sight, speed, and predation. The evolution of eyes, simple light-sensing patches evolving into complex image-forming organs, allowed organisms to see and be seen. Predators hunted with purpose. Prey developed defenses. The arms race of evolution accelerated, spurring rapid diversification. Hard shells, exoskeletons, spines, and camouflage evolved in response to ecological pressures. Life had become dynamic, interactive, and richly interdependent. Continents shifted, climates changed, and oceans transfigured themselves over eons. But life persisted, exploring new avenues of form and function. Plants ventured onto land, bringing with them the oxygen they had produced in the seas. Fungi followed, breaking down rock and preparing soil. Arthropods crawled from tidal pools, and vertebrates, those with internal skeletons, soon trailed behind. The Earth, once dominated by single-celled life, had become a theater of ecosystems, a planet teeming with sensation and response. Yet even in this riot of diversity, the question lingered in every strand of DNA. How had it all begun? The answer echoed in the chemical conversations of RNA, the membranes of protocells, the engines of mitochondria, and the quiet code of genes passed down across eons. From the sulfurous breath of vents to the symphony of the rainforest, life remained a singular expression of Earth's ancient chemistry made animate. The story, though ancient, was not finished. Evolution continued its work with patient inevitability. Some lineages would flourish, others would vanish. Mass extinctions would clear the stage, new forms would emerge from the ashes, and somewhere, in the branching complexity of life's tree, a creature would one day rise that could reflect upon the journey, could chart the past not by instinct, but by thought. That creature, us, was still far in the future, but the foundations were already laid. The patterns of behavior, the drives for survival and reproduction, the sensory frameworks and adaptive strategies, all were forming in those early multicellular organisms. Life was becoming aware, not yet of itself, but of its environment. It could now chase, flee, sense warmth and shadow. The world was no longer blind. Evolution had opened eyes. And so, the slow symphony of beginnings played on, its themes echoing. In coral reefs and forest canopies, in microscopic plankton and soaring birds, what started as a ripple in a chemical tide had become a planet alive with motion, color, and desire. And all of it, every cell, every wing beat, Every whisper in the dark was a continuation of that first molecular breath beneath the ancient sea. The air, once laced only with the mist of oceans, began to thicken with the breath of a new green world. Tiny pioneers, algal mats, and simple moss-like plants crept from the shallows of primordial ponds onto damp stones, forging the first footholds beyond the sea. Their delicate fronds captured sunlight and exhaled oxygen, transforming barren rock into nascent soil. In their wake, fungus and bacteria formed symbiotic partnerships, weaving networks of decomposition that would become the foundation of terrestrial ecosystems. Earth's dry lands, once desolate, now offered living shelter. As soils deepened and landscapes softened, the ancestors of fish felt the pull of a boundless frontier. Fins thickened into lobes, muscular flaps pressed upon shallow waters. Between, tides beneath the hiss of long-vanished storms, these pioneering vertebrates gulped air, their gills adapting to lungs, primitive bellows for life above the brine. These early tetrapods ventured onto mud flats, webbed feet leaving tracks destined to become fossilized records millions of years later. The thrill of movement on solid ground, unbroken by currents or waves, unlocked new modes of feeding, new niches in shadow and light. Forests of horsetails, club mosses, and primitive ferns rose behind them. Towering tree-like plants, some exceeding 30 meters in height, colonized riverbanks and swamps. Coal forests spread across continents, their decaying remains destined to fuel the fires of distant eras. Within the tangled undergrowth, invertebrates bloomed, giant millipedes, armored arthropods, and early insects that scuttled through leaf litter, their exoskeletons echoing the strength of innovation. Wings unfurled in some lineages, giving birth to the first aerial explorers. Their fragile flight and instinctual dance between gravity and the unseen currents of wind. Predators stalked these primeval woods. Early amphibians, slender and toothy, slithered along banks and lunged at unsuspecting prey. Their skins, still dependent on moisture, bound them to humid hollows, yet they thrived in the hush beneath fern canopies. Through countless generations they refined their limbs, their vertebrae, and their senses. Eyes rotated to the top of their skulls, nostrils edged the snout, and ears evolved to pick up vibrations in the air. Life on land demanded new architectures of body and mind. Then came the age of reptiles, a lineage that broke free from the watery tether by enclosing its eggs in leathery capsules, impervious to drying. These shelled embryos could be laid on sun-warmed ground, opening vast landscapes for colonization. With the innovation of the amniotic egg, vertebrates no longer required swamps for reproduction. They spread into deserts, highlands, and forests alike. Scales replaced slimy skin, providing protection and water retention. Efficient lungs and a more advanced heart enabled greater endurance and activity under the blazing sun. Under open skies, reptilian giants emerged. In the Mesozoic world, dinosaurs of every stripe and scale stamped the earth with their immense bulk and daring anatomy. Long-necked sauropods grazed the treetops. Swift theropods hunted in packs. Armored ankylosaurs bristled with defenses. Crested hadrosaurs trumpeted in herds. Their bones speak in silent testament to a reign that would last over 150 million years. Through volcanic winters, asteroid disruptions, and shifting continents, dinosaurs dominated every terrestrial realm. Yet life is shaped by change, and change arrived in cosmic fury. 66 million years ago, a scar appeared in the sky, an asteroid's final gift. The impact plunged the world into darkness, cold, and chaos. Forests burned, seas acidified, and the dominant reptiles perished in convulsions of extinction. But extinction is also opportunity. In the aftermath, the survivors, small, nocturnal, furry mammals, slipped from shadows into open niches. With dinosaurs gone, mammals radiated across the globe. Early mammals were modest creatures, but their warm-blooded metabolism granted them active lives in diverse climates. Hair provided insulation, and differentiated teeth allowed varied diets. Some became insectivores, others gnawed seeds, some took to the trees. Within their ranks, primate ancestors grasped branches with grasping digits. Their eyes rotated forward for depth perception, and their brains began to expand. These arboreal primates glimpsed patterns of light and shadow, subtle motions of leaves and limbs, forging the neural circuits that underlie curiosity. On the forest floors below, flowering plants, angiosperms, exploded into diversity. Their petals glowed in vibrant hues to lure pollinators, insects, birds, even small mammals. This intimate dance of flower and visitor accelerated evolution on both sides. Pollinators honed delicate adaptations, proboscises, beaks, fur, for precise flower visits. Flowers, in turn, refined their shapes and scents. A co-evolutionary tapestry wove itself across waterways and valleys. With abundant fruit and seeds, new opportunities emerged. Some mammals ventured outward, colonizing grasslands, tundras and deserts. Hoofed ungulates galloped across plains, their herds reshaping soil and vegetation. Carnivores, dogs, cats, bears, evolved specialized teeth and claws, mastering pack tactics and solitary stealth. A new era of ecological complexity burgeoned. Ecosystems of grass, herd, predator, scavenger, and decomposer formed dynamic balances beneath open skies. In some pockets of forested refuge, hominid lineages took their first faltering steps. Bipedal posture freed the hands for toolmaking. Simple stone flakes cut meat and plants, enabling larger brains through higher quality diets. Hands that once clung to branches now manipulated objects with precision. The spark of technology ignited neural growth. Neural growth fueled more sophisticated tools. A feedback loop between mind and culture pressed human evolution forward at unprecedented speed. Though true humanity lay far ahead, the groundwork was laid in these woodland clearings, social bonds, cooperative foraging, and the beginnings of language, a code not yet written on papyrus or silicon, but expressed in gestures, calls, and shared attention. Life, in its grand experiment, had achieved a new form of reflection, awareness of self in relation to community and environment. From the drying mud of ancient pools to the first flickers of consciousness, the journey of life on Earth had spanned eons, shaped by chance, physics, chemistry, and relentless adaptation. Continents shifted, climates oscillated, and cosmic impacts scarred the planet, yet life persisted, diversified, and thrived in forms we are only beginning to fathom. The tapestry of existence continued to expand, each thread a story of mutation, selection, and boundless creativity. The fire of thought had only just begun to smolder within the quiet minds of early hominids, but its warmth would one day light the world. On the plains of Africa, under skies arched with relentless sun and star-swept silence, beings emerged who stood more upright than their ancestors, whose hands carried not just tools, but intention. Homo habilis struck stone against stone, coaxing sharp flakes from ancient cores, and in the rhythm of those sparks came the first echoes of foresight. The hands that once clutched fruit or fumbled branches now shaped the very environment, drawing survival not from reaction, but from planning, from choice. From habilis to erectus, the lineage marched through time. Homo erectus, tall and lean, migrated far beyond ancestral homelands. They crossed thresholds of geography, of temperature, of fear. Fire came into their possession, not as lightning's fleeting touch, but as a lasting guardian in the night. Flames danced in caves and beneath overhangs, casting shadows of bone and fur, warming flesh and warding off the eyes that glowed in the dark. Cooked food softened the burden on the digestive tract, and energy turned inward, toward brain, toward vision, toward dreaming. And dream they must have, for language was not far behind. Though no record exists of their first words, no script, of their earliest murmurs, sound began to acquire meaning beyond warning or mating call. A call could carry memory, a tone could signal future intent. In time, meaning poured into syllables, and with it the bond of shared mind. Language transformed the brain into a vessel not just for thought, but for the transmission of thought. An echo passed from one life to another, surviving beyond the flesh. Homo sapiens emerged from this lineage like a spark catching dry tinder. With rounded skulls and high foreheads, they carried not just new genes but new ideas. Their tools bore elegance. Blades flaked with geometric precision. Spears waited for flight. They carved bone, painted caves, and adorned themselves with ochre and bead. In these acts of symbolic creation, life announced that it now recognized itself. The earth, so long governed by chemical reactions and natural selection, now sheltered creatures who wondered why. Migrations swept the globe. Sapiens crossed rivers, deserts, and frozen straits. Each step forward wove the human story. Into the landscapes of Asia, Europe, Australia, and the Americas. They encountered new species, Neanderthals, Denisovans, and interbred, their bloodlines blending and braiding the story of ancestry. In each region, adaptations sculpted bodies to match the land. Skin darkened or lightened, nostrils widened or narrowed, all in quiet negotiation with the climate, the altitude, the sun. The diversity of culture bloomed alongside the diversity of biology. Language diverged into dialects and then into families. Customs rooted in ecology blossomed into religion, into ritual, into myth. Each tribe held its stories of sky gods and water spirits, of ancestral beasts and sacred hills. Around fires beneath stars, people imagined how it all began. They told of cosmic eggs, of serpents in trees, of gods breathing dust to life. These were not just stories of origin. They were frameworks for identity, maps to navigate the intangible terrain of wonder. And beneath the myths, science waited, not as a rival but as a different lens. Some asked, not only who made the stars, but what are the stars? Not only who shaped man, but how was man shaped? These questions, carried like seeds through countless millennia, finally found root in minds unafraid to ask more than they could answer. Observations began to eclipse tradition. The flicker of logic appeared. Evidence became holy in its own right. The birth of agriculture reshaped life's pattern once more. Where once bands wandered with game, now they stayed and sowed. Wheat and barley, millet and maize rose from domestication to sustain civilizations. With steady food came surplus, and with surplus came hierarchy. Cities unfurled along rivers, Tigris, Nile, Indus, Yellow, and writing emerged to track their rise. Cuneiform tablets and hieroglyphic scrolls bore the imprint of a mind turned toward permanence. What was once spoken could now be preserved across centuries. Within these cities, minds turned skyward. Priests charted the heavens, scholars tallied eclipses. From Babylon to Alexandria, theories of creation mingled with mathematics. Thales guessed that everything was made of water. Anaximander proposed boundless beginnings. Empedocles named 4 elements: earth, air, fire, and water. But none could yet grasp the deeper machinery of life itself. Still, questions lingered like constellations overhead. How did life begin? Was it always here, or did it arrive from elsewhere? Could a divine breath spark flesh, or could matter itself awaken under certain conditions? With each passing century, knowledge accumulated like sediment at a river delta. Some believed in spontaneous generation, that life burst from non-living matter as easily as frogs from mud. Others doubted, questioning what was unseen. The microscope shattered the veil. In the 17th century, Antony van Leeuwenhoek peered into water drops and discovered a hidden world. Animalcules, tiny swimmers invisible to the naked eye. This was life at its most elemental, a teeming universe within a spoonful of pond water. Life, it now appeared, came in forms beyond imagination, each one governed by principles yet to be understood. The centuries marched on. Francesco Redi challenged the notion that maggots emerged from rotting meat without cause, sealing meat away, and watching the absence of life. Louis Pasteur would later drive the nail deeper, proving that even the air held secrets, microbes, spores, invisible architects of decay and fermentation. Spontaneous generation crumbled. Life did not erupt without cause. But how, then, did it begin? Chemistry rose to meet the riddle. Scientists dissected cells, identified proteins, lipids, sugars, and nucleic acids. In the nucleus of cells, they discovered chromosomes, twisted ladders of inheritance. The code of life hid within DNA, a molecule whose spiral arms carried the instruction for building every living thing. But DNA did not solve the origin. It raised new questions. How could such a complex molecule arise from chaos? Stanley Miller and Harold Urey stepped forward with an experiment that echoed through the annals of science. In 1953, they recreated Earth's early atmosphere, methane, ammonia, hydrogen, and water, and sparked it with electric arcs to mimic lightning. After days of boiling and zapping, the flask darkened. Within, they found amino acids, the building blocks of proteins, forged from lifeless elements. It was a revelation. Life's ingredients could emerge from simple chemistry. But ingredients are not recipes. And so the search deepened. Researchers explored the idea of protocells, tiny bubbles formed from lipids, capable of trapping molecules inside. These vesicles could replicate, split, absorb nutrients. Could life have started this way? Perhaps deep in the ocean, where hydrothermal vents gushed minerals and heat into the darkness. Ancient chemistry gave rise to metabolism before replication. Or perhaps on sunlit shores, drying and wetting cycles coaxed molecules into longer chains, like primitive scripts trying to write their first sentence. Even RNA, once thought merely a messenger, stepped into the spotlight. It could both store information and catalyze reactions. Was RNA the first self-replicating molecule? The RNA world hypothesis suggested that before DNA, before proteins, strands of RNA might have danced through primordial soups, making imperfect copies of themselves, weaving error and evolution into the earliest form of memory. But questions persist. Could life have come from the stars, hitching a ride on meteorites? Organic molecules have been found on asteroids, and amino acids rained down from space in the Murchison meteorite. Could panspermia, a seeding of Earth by interstellar dust, have been the match that lit the terrestrial flame? Answers remain elusive, yet each theory carries fragments of truth. In the oceans of Enceladus, the methane of Titan, the ancient rocks of Mars, and the interstellar ice of comets, we search for parallels. Because to understand how life began here is to ask if it could begin elsewhere. And in doing so, we seek not only to know our origin, but to map the potential kin of Earth across the vast quiet of space. Beneath the surface of our understanding, deeper questions stirred. If amino acids could form under prebiotic conditions, if lipid vesicles could arise spontaneously, if RNA could store and transfer information, then where was the threshold, the singular breath, where chemistry crossed the line into biology? Was there a moment when a soupy collection of molecules suddenly became something alive? Or did life arrive gradually, like dawn overtaking night? Nature does not always offer clear demarcations. The earliest forms of proto-life may have blurred the boundaries between animate and inanimate, between reaction and response. Within microscopic chambers, perhaps in mineral pores near hydrothermal vents or tide pool crevices laced with heat and salt, simple molecules began to interact in ways that echoed life. Feedback loops formed, metabolic pathways took root, and, from this fragile choreography of matter, rudimentary systems emerged that consumed energy, reproduced imperfectly, and evolved. These early molecular entities lacked cells in the way we know them now. They had no membranes, no genetic code, no organelles. Yet they exhibited a kind of behavior, simple, patterned, consistent. They grew and split, shifted and recombined. Some configurations endured longer than others. The environment tested them not with cruelty, but with indifference. And those that persisted slowly assembled complexity through chance and time. In this crucible of persistence, the first self-replicating systems took hold. If RNA was indeed the architect of this first inheritance, it must have possessed qualities of elegance and resilience. In the folds of its single-stranded spiral, RNA could assume shapes that catalyzed reactions, allowing it to direct chemical traffic while carrying the script of its own replication. Some strands copied themselves more efficiently, others folded into enzymes, ribozymes, that could influence surrounding reactions. Eventually, the dance of molecules led to partnership. RNA may have joined forces with peptides, short chains of amino acids, to stabilize its structures, protect its codes, and hasten its functions. This proto-metabolism, fragile and improbable, was enough to tip the balance. A replicating system with selective advantage had emerged. Mutation played its role, not as error, but as necessity. With variation came evolution. With selection came adaptation. From this seed of replication, the next leap arrived. Enclosure. A lipid bilayer, spontaneously forming spheres in water, could have surrounded replicating RNA and its allied molecules. Within this boundary, a microcosm emerged, protected. From the chaotic external soup, concentrations of molecules could rise, reactions could occur more efficiently, and natural selection could act more decisively. These were protocells, not yet full cells, but coherent systems on the brink of autonomy. Over time, membranes became more than walls. They became interactive thresholds, letting certain molecules in and out, responding to osmotic pressures, internalizing nutrients. The protocell became the platform for competition and innovation. Molecules that improved stability or replication conferred advantage. Internal structures evolved, enzymatic pathways, primitive cycles of energy conversion, rudimentary signal, transduction. And as these protocells multiplied, evolved, and occasionally fused, something extraordinary happened. A genetic code emerged, a standardized system for translating nucleic acid sequences into amino acids. This was the birth of proteins, complex molecules with diverse functions, each folded into specific shapes, each capable of catalyzing distinct reactions. With proteins came specialization. The once chaotic internal soup of the protocell became an organized network of tasks and efficiencies. At the heart of this machinery, DNA entered the scene. More stable than RNA, it began to take over the role of long-term information storage, while RNA became a messenger and facilitator. The central dogma of molecular biology formed. DNA makes RNA makes protein, and with this trinity, life as we understand it began to resemble its modern form. The first true cells, prokaryotes, took shape, their circular genomes spinning within the confines of lipid membranes. They were alone on a vast, hostile world, yet they were many. These early cells consumed the resources of their environment, adapted to extremes of heat, salinity, acidity, and radiation. They divided, mutated, endured. Some learned to harvest energy from chemical gradients in the Earth's crust. Others, in a leap of genius written in the language of molecules, discovered photosynthesis. They captured sunlight, split water, and released oxygen. The great oxygenation event began, not as an explosion, but as a slow, relentless flood. Oxygen, once rare and reactive, became abundant in Earth's atmosphere. It transformed the planet's chemistry, rusting iron into red bands in ancient rocks, triggering die-offs among anaerobic life forms, and opening new doors for metabolic innovation. Oxygen allowed for more efficient energy production through aerobic respiration. Life could now grow larger, faster, more complex. In this changing world, cells continued to evolve. One branch led to the archaea, organisms of extremes thriving in boiling springs and salt flats. Another became the bacteria, diverse and ubiquitous, present in soil, water, and every crevice of life. And then, in a singular act of cooperation, something new emerged, the eukaryotic cell. It began with one cell engulfing another, not to digest it, but to shelter it. The engulfed cell became a mitochondrion, an energy factory. A partnership became permanent. The eukaryotic cell was born. This new architecture allowed for compartmentalization, distinct organelles with distinct tasks. Nuclei held DNA, endoplasmic reticula synthesized proteins, Golgi bodies sorted and shipped. These cells grew larger, more versatile. They formed colonies, then filaments, then tissues. Some adhered together for safety, then evolved communication. Differentiation followed. Cells that moved, cells that sensed, cells that fed, cells that reproduced. From one came many, each with purpose. Multicellularity had arrived. At first it was simple, clusters of identical cells dividing and drifting, but in time, coordination grew. Genetic programs turned on and off in waves, sculpting embryos from zygotes, shaping organs from undifferentiated masses. Life began to fold itself into increasingly intricate patterns: worms, corals, fronds, and tentacles. From these humble shapes emerged creatures with nervous systems, eyes, limbs, and thoughts. The Cambrian explosion was not just an eruption of diversity, but of possibility. Within the oceans bloomed ecosystems of extraordinary variety: predators with complex jaws, swimmers with articulated spines, burrowers, grazers, filter feeders. The gene regulatory networks that once guided the formation of simple bodies now built shells, fins, antennae, and claws. Eyes evolved independently across lineages, sensing light in myriad ways. Brains began to interpret the world, not just react to it. Life was no longer merely enduring, it was expressing itself. Evolution became not just a response to pressure, but a sculptor of form, a composer of beauty. From a spark in ancient chemistry arose a planet teeming with vision, motion, and desire. And through it all, the trace of the beginning remained, hidden in the genomes of every living thing. Within each cell, mitochondria still carry their separate DNA, a relic of their bacterial ancestry. Ribosomes still read RNA in a language billions of years old. The code of life, though evolved and reshaped, still bears the mark of its primordial roots. Every breath, every heartbeat, every thought is a continuation of that first flicker of animation in Earth's early oceans. The origin of life is not a single point on a timeline, but a cascade of thresholds crossed one after another, from molecule to machine, from chaos to consciousness. And though we search for a singular genesis, we find instead a tapestry woven from countless accidents and epiphanies, each one building upon the last, each one etched into the memory of matter. The journey from chemistry to biology is the journey of becoming. It is the deepest story we can tell, one written not only in books, but in blood and breath, in DNA and stars. And still, it is not finished. Life continues to unfold its potential, to explore its limits, to dream. In the quiet pulse of Earth's ancient oceans, life advanced by layering upon its own innovations, building slowly from simplicity to intricacy, never abandoning what had worked, but instead reconfiguring it for new terrain. The eukaryotic cell, once a singular revolution, became the platform from which entire kingdoms of life emerged. Plants, animals, fungi, all descending from common ancestry but each diverging like branches twisting from the same root. The mechanisms that powered this divergence were not orchestrated but emergent, born from mutation and filtered by the sieve of natural selection. Genes duplicated, errors crept into strands of DNA, recombinations shuffled instructions, and with every replication, the code rewrote itself. Most of these changes vanished into oblivion, but some endured, and those few drove the arc of evolution forward, relentlessly and blindly. Multicellular organisms began to dominate not because they were fated to, but because they could do what no single-celled organism could: divide the labor of living. A multicellular body could afford specialization. Some cells could gather light, others could process nutrients, others could build structure or sense the world. From these distributed responsibilities came tissues, organs, systems. Life now had directionality, growth stages, reproduction strategies, defense mechanisms, and mobility. Among the earliest multicellular pioneers, algae claimed the sun. They transformed oceans into cradles of oxygen, contributing to the atmospheric bloom that fueled the next wave of complexity. These photosynthetic organisms gave rise to the first plants, which would later crawl onto land, blanketing the barren continents with green, ferns, and mosses, towering horsetails and cycads. They sculpted the landscape, shaping not only the air but the soil itself through root systems that fractured rock and funneled nutrients. But even before the land was conquered, another revolution stirred underwater. Among the creatures of the Cambrian seas, certain forms began to favor symmetry and segmentation. This architectural innovation allowed for greater movement, coordination, and sensory capacity. With segmentation came the potential for limbs, exoskeletons, antennae, and other elaborate adaptations that allowed organisms to explore their environments with unprecedented sophistication. From this lineage arose the arthropods, trilobites, that scuttled along sea floors, ancestors of insects and crustaceans that would one day crawl across land and sky. In another branch, the chordates evolved, animals with a notochord, a primitive backbone that later stiffened into vertebrae. These creatures would become fish and later all tetrapods, amphibians, reptiles, birds, and mammals. As vertebrates proliferated, their internal scaffolding allowed for larger bodies, stronger musculature, and more intricate nervous systems. Vision sharpened, balance improved, sensory systems diversified. Fins became limbs, lungs emerged from swim bladders. And with gradual adaptation, the first vertebrates ventured from sea to shore, bringing life with them into a new domain. The colonization of land was no small feat. Gravity pulled harder. Desiccation threatened every cell. Reproduction could no longer rely on aquatic dispersal. Yet life responded not with hesitation, but with experimentation. Amphibians lived half in water, half on land, serving as transitional forms. Reptiles developed eggs with protective shells, liberating them from aquatic reproduction. Scales retained moisture, lungs grew more efficient, and bodies adapted to the dry pulse of terrestrial cycles. In time, the earth bloomed with reptiles, sprawling across continents, diversifying into forms both monstrous and miniature. Dinosaurs thundered across floodplains and forests, some taking to the skies with wings of membrane or feather. Their reign lasted tens of millions of years, an empire of blood and bone, of roars and silent stares beneath alien stars. And yet, like all empires, it ended. A cataclysm from the cosmos, a meteorite or comet struck Earth with fire and force, shrouding the planet in dust, igniting a long extinction. The dinosaurs fell, but from their absence emerged opportunity. Mammals, once scurrying in the shadows, rose into the light. They expanded into every niche, gliding, swimming, galloping, climbing, guided by warm blood and complex behaviors, by internal gestation and parental care. Among these mammals, the primates began to rise. They grasped with hands, balanced in trees, saw in color, communicated through gesture and sound. Social bonds grew strong, learning passed from one generation to the next. In one lineage, bipedalism emerged. The hands were freed, the skull balanced above the spine, and brain volume began to expand. This was the birth of hominins, a journey that would unfold across the Rift Valley of Africa, across millennia of change, pressure, and promise. Fire was tamed, tools were shaped, and stories began to pass not through DNA, but through memory. Mouth. Homo habilis gave way to Homo erectus, who journeyed from Africa into new lands, their footprints tracing the first great migration of consciousness. Eventually, Homo sapiens appeared. They carried the weight of ancestors and the potential of worlds. With language, they could pass wisdom. With imagination, they could reshape their environment. With collaboration, they formed societies. With wonder, they looked up at the stars and asked, "Where did we come from?" And yet their story was still the same story. The extension of a single line traced back to that first replicating molecule in Earth's primordial tide. Everything about us, our cells, our bones, our minds, is written in the ink of eons. Our DNA still speaks the tongue of ancient microbes. Our breath echoes the rhythm of oceans long vanished. Our thoughts are shaped by the same laws of chemistry that gave rise to life from lifelessness. But the origin of life, as science seeks to understand it, is not simply about beginnings. It is about possibility. It is about emergence, how complexity arises from simplicity, how order can rise from randomness under the guidance of energy and feedback. The Earth, once silent, now hums with the voices of 10 million species, each one an iteration of the same cosmic experiment. From stardust to cell, from protocell to symbiosis, from microbe to mammal, from synapse to sonnet, the path was never linear, never inevitable, never simple. But it was guided by the quiet, persistent logic of nature, sculpting and discarding, refining and renewing. Life, then, is not an event. It is a continuum, a process, a whisper made louder through repetition and variation, a self-sustaining flame that carries forward the light of origin through every living thing. And though we have come far in our understanding, the ultimate question still shimmers like a mirage at the edge of knowing. Could it have happened differently? Could life have arisen again and again, only to be extinguished before it left a trace? Could the universe itself be seeded with the same potential, awaiting only the right moment, the right pressure, the right spark? As we turn our gaze to other worlds, Mars, Europa, Enceladus, exoplanets circling distant suns, we carry with us the insights of Earth's own genesis. We know what to look for: chemical disequilibrium, liquid water, complex organics, energy gradients. But we also carry humility, knowing that life, as we know it, is both common in its chemistry and rare in its resilience. We search not for answers, but for echoes, for signs that the universe has repeated the miracle we call life, for hints that the story of Earth is not an exception, but a stanza in a larger cosmic poem. And in that search, we are once again part of the story, not merely observers, but participants. The universe through us has become aware of itself. It dreams through us, questions through us, remembers through us. The origin of life is not behind us. It is within us. It is our inheritance, our identity, our fire. Beneath the crust of Earth and the corridors of the mind, the memory of life's origins remains etched in molecules, patterns, instincts, and cycles. Though humanity stands at the pinnacle of biological complexity, it is not separated from the primal forces that once brewed the first cell into being. Instead, it is a living extension of that same great unfolding, each heartbeat a continuation of the rhythm that began in an ancient tide pool beneath alien skies. What began as chemistry has become consciousness. The molecules that once merely floated and collided now ponder their own arrangement. The spiral of DNA, once merely a mechanism of replication, has become a mirror in which the universe glimpses itself. And as science deepens its reach, tracing backwards through fossils and isotopes, through simulations and spectrometry, a deeper portrait begins to emerge. A portrait not only of what life is, but of the cosmic conditions that make it possible. The universe, vast and cold, is governed by symmetry and law, yet amid its order lies the subtle imperfection from which diversity blooms. Stars ignite through nuclear fusion, but their deaths seed galaxies with the ingredients of life: carbon, oxygen, nitrogen, phosphorus, sulfur, iron. These elemental ashes, scattered by supernovae and cradled within interstellar clouds, give birth to new stars and planets, each a unique crucible of potential. Earth among them was not predestined but positioned, neither too close nor too distant from its sun, spinning with a tilt that orchestrates seasons, harboring a moon that steadies its axis, layered with a magnetic field that shields it from cosmic rays. Its surface cycles water between ocean and cloud. Its tectonic plates breathe carbon in and out. Its atmosphere retains warmth without suffocating heat. Here, complexity found a fertile stage, and from chaos, patterns arose. Yet even with perfect conditions, life did not burst forth as a singular, miraculous event. It crept, it experimented, it failed again and again. The fossil record is silent, on many of these failures preserving only the victors. But beneath that silence lies a truth. Evolution is not the story of steady improvement, but of branching possibilities, some explored and abandoned, some amplified and adorned. Molecular biology reveals that many of the tools of modern life, ribosomes, enzymes, proton gradients, originated in organisms long extinct. Horizontal gene transfer passed innovations sideways across species. Entire metabolic pathways were shared like communal inventions. Life was never solitary. It was communal from the start, a grand collaboration of parts finding synergy. From this collaboration emerged the cell's genius: compartmentalization. Membranes enclosed reactions, separating interior from exterior, building gradients, fostering complexity. With organelles came the ability to orchestrate energy with precision. Mitochondria generated ATP, chloroplasts captured photons, nuclei stored blueprints. Each organelle was a revolution in efficiency. Life no longer merely reacted, it planned, stored, adapted. And with genetic storage came memory, not memory of events, but of function. DNA does not recall days or dreams, but it remembers how to survive, how to fold a protein, how to recognize a toxin, how to grow a limb. Over billions of years, these molecular memories accumulated into the library of life, curated by trial and error, annotated by mutation, refined by selection. But if life is memory, then it is also forgetting. Extinction has claimed over 99% of all species to ever live. Their bones lie buried in strata, their stories hinted in isotope ratios and genomic ghosts. Each extinction a reset, each survival a lottery won. The Cambrian explosion dazzled with diversity. The Permian extinction erased most of it. The dinosaurs ruled, then vanished. The mammals rose not through conquest, but through absence, an empty stage inviting new actors. And in one corner of that stage, a new kind of mind emerged. Not just aware, but self-aware. Not just social, but symbolic. Language allowed thoughts to transcend time. Tools extended the hand. Fire softened the night. Art carved permanence into fleeting breath. Homo sapiens, inheriting a lineage 4 billion years in the making, began to shape not just ecosystems, but the meaning of existence itself. Still, our origins hold us. The architecture of our brains echoes the wiring of earlier vertebrates. The genes that guide our development once sculpted fish. Our microbiome contains the fingerprints of ancient symbioses. We are colonies within. Colonies, human in thought, microbial in number, stardust in composition. And the question persists: was it inevitable? Given the right conditions, would life always arise? Or is our existence a rare alignment, a cosmic fluke never to be repeated? The answers remain buried in probabilities and time. But by studying life on Earth, its beginnings, its constraints, its trajectories, we construct the lens through which to search for life elsewhere. In the icy plumes of Enceladus, where hydrothermal vents may churn beneath an ocean of darkness. In the reddish soil of Mars, where ancient rivers once cut through valleys. In the atmospheres of distant exoplanets, where strange chemistries hint at equilibrium disturbed. The search is guided not by hope, but by understanding. Astrobiology, once a dream, is now a discipline. It fuses biology, geology, astronomy, and chemistry, asking how matter becomes alive and where else it might have. It constructs synthetic cells, simulates early Earth environments, and deciphers the spectral fingerprints of distant worlds. It seeks not to replicate life, but to understand its essence, the delicate, persistent defiance of entropy through structure and purpose. In laboratories, protocells are coaxed into self-replication. RNA strands evolve through selection. Proteins fold in silico, learning to mimic the complexity nature achieved over eons. And in these pursuits, we glimpse not just the past, but the architecture of potential futures. What we find may not resemble life as we know it. It may not breathe oxygen or encode DNA. It may dwell in solvents we cannot drink, at pressures we cannot endure, under suns we cannot see. Yet if it sustains itself, reproduces, evolves, if it passes the torch of existence forward, It belongs to the same lineage of miracle. And so we return to the original question. How did life begin on Earth? We may never point to a single moment, a singular reaction, a single molecule, and declare, here, life began. But we can trace the converging paths of physics forming atoms, of chemistry building complexity, of biology emerging from self-replication, of consciousness rising from neurons sparking in the dark. It was not an event, but a dawn. Not a miracle, but a crescendo. Not a question of if, but of when. And perhaps that is the deeper answer, not in the mechanism, but in the realization that life is not foreign to the universe. It is an expression of it. When matter organizes itself under the guidance of energy and time, when feedback loops build resilience, when memory meets adaptability, life is not just possible, it is a natural consequence. In every cell that divides, in every breath that stirs the air, in every question we ask of the stars, the origin of life continues. We are not separate from that story, we are its newest verse. The night sky, scattered with stars like ancient embers, carries in its silence the same story written in the molecular whispers of our blood. The origin of life on Earth was never an isolated event. It was a cosmic unfolding. From the raw dust of galaxies to the fragile shimmer of cell walls, the emergence of life was not a spontaneous miracle, but the result of a universe experimenting with order in the midst of entropy. As we look deeper into the soil beneath our feet and the heavens above, we begin to sense the rhythm that links stars to cells, time to consciousness, gravity to desire. The language of life is not confined to biology alone. It is spoken by water as it carves riverbeds, by volcanoes as they spew mineral-rich breath into infant seas, by lightning that dances across primordial skies. All of these were actors in the opening scenes of Earth's great drama. The very rocks that cradle our modern cities were once molten, churning cauldrons where the elements for life were brewed, cooled, and solidified into invitation. In these ancient crucibles, the elements bonded and broke, seeking stability in ever more complex forms. Carbon, with its 4 arms, became the great architect of diversity. Hydrogen danced with oxygen to pour the world's oceans. Nitrogen lent its strength to the scaffolding of genes. Every atom that life would later need had been forged in stars and strewn by their violent deaths, scattered like seeds across the galactic field. Yet even with the right ingredients, the recipe was not immediate. For hundreds of millions of years, the Earth simmered in a volatile quiet. Comets and asteroids battered its surface, delivering water and organic molecules, but also extinction in their wake. Volcanoes belched gases that thickened the atmosphere. Solar radiation scorched the surface, unfiltered by an ozone layer that had not yet been born. Still, the Earth endured, and in its resilience, conditions began to shift. Oceans gathered, continents cooled. The atmosphere thickened with carbon dioxide and methane, forming a greenhouse that trapped the sun's warmth. Life needed only the right moment. And in that moment, something extraordinary began. A handful of molecules, perhaps on the edge of a hydrothermal vent or within a mineral crevice, began to copy themselves. These early replicators, not yet life as we know it, were blind but persistent. Some copied with greater stability, some with greater speed. And with each iteration, evolution began its slow, implacable work. Mutations introduced variation. The environment imposed selection. Molecules that could copy more reliably began to dominate. This was not yet life, but the prologue of its symphony. With time, the simplicity of self-replication gave way to competition. Molecules began to interact, to form networks where one's survival depended on the function of others. Feedback loops emerged. Chemical gradients were harnessed to fuel reactions. Inorganic catalysts, clays, metals, may have offered scaffolds for these reactions, lending the process both stability and direction. The stage was set for something more structured, more intentional. The cell. The first protocells were likely flimsy spheres of lipid membranes, floating in water like bubbles. Within them, primitive reactions were shielded from the chaos of the outside world. These enclosures allowed concentration, protection, and eventually cooperation. Inside, RNA may have both stored information and acted as a catalyst, the dual role of script and performer. Over time, proteins, more versatile and precise, took over catalysis. DNA, more stable than RNA, became the archive. The Great Division of labor had begun. These first cells were anaerobic, living in a world without oxygen, feeding on the chemical richness of their environment. But change is inevitable. As some cells evolved the ability to harness sunlight through a primitive photosynthesis, they began to exhale oxygen as waste. What was poison to one became possibility to another. Oxygen built up in the oceans, then the skies, transforming the Earth in what scientists now call the Great Oxidation Event. This oxygenation was both apocalypse and rebirth. Many ancient life forms perished, but others, through innovation, learned to use oxygen in their metabolism, unlocking far greater energy than fermentation alone could provide. Energy is possibility, and with it came the ability to grow, to adapt, to explore new forms. Complexity blossomed. Around this time, another miracle of biology occurred: endosymbiosis. A larger cell engulfed a smaller one, not to digest it, but to live in partnership. This internal companion became the mitochondrion, the powerhouse of modern eukaryotic cells. Later, in plant ancestors, another engulfed cyanobacteria, giving rise to chloroplasts. These symbiotic events were not mere accidents. They were evolutionary leaps, joining formerly separate life forms into unified wholes more powerful than the sum of their parts. With eukaryotic cells came organelles, compartments of specialized function. These cells could now manage internal environments with greater precision, grow larger, store more genetic material, and eventually specialize. From this versatility emerged the first multicellular organisms, cooperating collections of cells that could form tissues, organs, bodies. Life had crossed another threshold. Still, for billions of years, life remained microscopic, hidden from the naked eye. Only in the Cambrian explosion, roughly 540 million years ago, did complexity erupt visibly. Fossil records show an astonishing diversification of body plans, many of which persist today. Eyes, limbs, shells, and nervous systems evolved in dazzling variety. Evolution was no longer creeping, it was cascading. The Earth, shaped by plate tectonics, climate cycles, and cataclysms, was a constant agent of change. Extinctions cleared the slate, and evolution filled it anew. From water to land, from fin to limb, from gill to lung, life adapted. The carbon pulse of forests, the predatory elegance of dinosaurs, the icy patience of glacial ages, each chapter of Earth's history wrote itself in the bodies of its creatures. And then, from within this procession of forms, emerged beings who could remember not only with genes, but with stories. They painted caves with the memory of hunts. They fashioned tools that outlasted their hands. They buried their dead, wondering perhaps what breath was and where it went. Human beings, shaped by the same molecules as all life before them, became historians of the Earth itself. But to understand where we go, we must continue to ask how we began. The question of life's origin is no longer confined to biology. It touches geology, physics, astronomy, and philosophy. Each discovery draws the veil back a little further. Each simulation, each analysis of meteorites, each exploration of the deep sea or the Martian soil is a step toward the beginning. We may never find a single answer. There may have been many paths to life, some now lost to time, others echoed in the genes of modern microbes. But every clue, every nucleotide, every isotope ratio, every fossil, is a note in the cosmic melody that gave rise to thought, feeling, motion. And still, life presses onward. It adapts to radiation, to extreme pressure, to boiling acid. It flourishes. In the darkness beneath Antarctic ice, in the sulfurous plumes of undersea vents, on the desolate surface of the stratosphere. Life's persistence is not merely stubborn, it is transcendent. It is the echo of a universe that, against all odds, has chosen to know itself through biology. As we continue to study this origin, we study ourselves, not just our past, but our place in the cosmos. The atoms in our hands were once in the hearts of stars. The enzymes in our cells whisper of ancient oceans, and the questions we ask are written in the same language as the code that built our minds. In the silence that followed the first replication, a rhythm began. It was not yet life as we understand it, but it was motion, repetition, emergence. With every cycle, the raw materials of Earth, its bubbling vents, sunlit shallows, and mineral-rich basins were becoming theaters for a molecular symphony. Carbon chains bent into spirals and lattices. Simple sugars danced with phosphate backbones. Through trial and error, the molecules of non-life were sculpting blueprints for living things. This was not a planned design, but a chemical inevitability. The Earth in its early age was not merely a passive host but a catalyst. Its oceans, salty and warm, churned with potential. Its crust, fractured and porous, offered both shelter and fire. Its atmosphere, laced with lightning and ultraviolet light, energized the improbable. Within this restless world, the code of life began to inscribe itself, not on paper but in strands, chains, loops, in the spinning elegance of spirals unseen to the naked eye. Among the molecular players, RNA began to assert dominance. Its structure, simple yet functionally diverse, enabled it to both carry information and catalyze reactions. This duality placed it at the center of what some call the RNA world, a proposed stage in the origin of life where RNA molecules formed, replicated, and evolved. Though more fragile than DNA, RNA had the advantage of versatility. It could mold itself into shapes that performed tasks, cutting and joining, copying and folding. As RNA systems became more intricate, stability became necessity. DNA emerged as a more durable archive, taking over the task of long-term information storage. Proteins, composed of amino acid chains, arose as superior catalysts. Their diversity of shape allowed for exquisite specialization, enzymes that could accelerate life's chemistry by factors of millions. Thus, the division of life's three fundamental roles, store, transmit, catalyze, was complete. DNA remembered, RNA delivered. Proteins built. At the same time, the cell, the smallest unit of life, had begun to evolve its identity. Encased in membranes formed from lipids, these primordial cells regulated what came in and what went out. This boundary was not just physical, but informational. Within the membrane, a world of reactions could unfold, protected from external chaos. The lipid bilayer, self-assembling and selectively permeable, became the fortress of life, a place where complexity could flourish in isolation. Inside, a dance of metabolism began. Energy, the currency of all activity, was captured, transformed, and spent. Early cells likely fed on the bounty of Earth's natural gradients, chemical differences between hot and cold, acidic and alkaline, light and dark. But as resources became scarce, the pressure to innovate grew. Evolution demanded more efficient pathways, more cooperative molecules, more resilience in the face of change. Some cells discovered photosynthesis, not the refined oxygen-producing form of today, but a cruder version that harnessed sunlight to split molecules and create energy-rich compounds. Others evolved chemosynthesis, using inorganic molecules like hydrogen sulfide to fuel their processes. These discoveries expanded life's reach, allowing it to flourish in sunlit tide pools and pitch-black ocean vents alike. The accumulation of oxygen in the atmosphere, born from advanced photosynthesis, marked a turning point. Oxygen, once a trace gas, became a force. It reacted violently with many substances, breaking down unprotected molecules. For countless organisms, it was lethal. But for others, it was liberation. Aerobic respiration, which uses oxygen to produce energy, unlocked vast reserves of power. Life could now support larger bodies, faster movement, and more intricate internal machinery. This surge in energy availability set the stage for the next revolution, the rise of eukaryotic cells. These were not merely bigger cells, but structurally more sophisticated. Inside them, membranes folded into organelles, each with its task: power generation, protein synthesis, waste management. Mitochondria, ancient bacteria that took up residence within larger cells, became indispensable partners. Their story is one of symbiosis, a living alliance that allowed both entities to survive and evolve. The genome of the host cell and that of the mitochondrion merged slowly, gene by gene, into a new narrative. This cooperation, encoded in DNA and perpetuated in every cell division, was a leap not just in biology, but in logic. Life was no longer isolated individuals, but networks of interdependence. It was a principle that would echo across all future evolution. As eukaryotic cells diversified, they explored new forms. Some remained solitary, thriving in isolation. Others began to aggregate, first loosely, then with increasing integration. Over time, specialization took hold. Some cells in a colony might focus on movement, others on nutrient absorption, others on reproduction. From this differentiation came tissues, layers of coordinated function, and from tissues, organs, from organs, organisms. Multicellularity did not arise once, but many times in different lineages. Algae, fungi, plants, and animals all took their own paths toward collective living. Each lineage developed ways to control growth, communicate between cells, and regulate the death of parts to sustain the whole. These strategies laid the foundation for the complexity that now fills forests, oceans, and skies. With multicellularity came time. Longer lifespans, slower reproduction, more intricate development. Organisms could grow large, adapt more precisely, and pass along not just genes but behaviors. The nervous system began as a simple web of communication, a means to detect danger or light. But in animals, it blossomed into a command center. Signals traveled through networks of neurons. Movement became purposeful. Senses evolved. Eyes to see, ears to hear, tongues to taste the chemical world. Every sensation was another layer of interaction with the environment, another opportunity to thrive or fail. Evolution, relentless and impartial, refined each ability. Over millions of years, the pressure to survive shaped wings and fins, claws and camouflage, venom and memory. Through natural selection, life diversified into the bewildering richness we now observe. But alongside these bodily transformations, something subtler was unfolding: consciousness. In animals with increasingly complex brains, behavior became more than reaction. It became choice. Awareness flickered in the minds of creatures that could feel pain, anticipate threats, remember locations. Though its origins are still mysterious, consciousness is now seen not as a sudden appearance, but as a gradient, a continuum stretching from the simplest sensations to self-reflection. In one branch of this tree of life, primates evolved. Among them, a group of upright walking apes began to shape their world with more than instinct. They used tools, they shared food, they painted symbols on stone. Homo sapiens, the storytellers, the question askers, the fire keepers, stood at the junction of biology and narrative. In their language and technology, the origin of life gained a voice. That voice now wonders: was life on Earth inevitable given the laws of physics and chemistry? Or was it an improbable bloom in an otherwise indifferent universe? This question drives the search beyond our planet. In the icy moons of Jupiter, in the ancient sediments of Mars, in the atmospheres of exoplanets light years away, we seek echoes, molecular hints that life is not unique to us. Astrobiology has become the extension of our quest to understand how life began. It studies extremophiles, organisms that thrive in boiling acid or freezing salt, to infer what might live elsewhere. It analyzes meteorites for amino acids. It simulates early Earth conditions in labs to recreate the spark of biogenesis. Every experiment brings us closer to seeing the full picture, the universal contours of life's possibilities. But even as we look outward, we must not forget the inward journey. For the molecules that began this story are still within us. The RNA that once ruled alone still serves. The proteins that built the first enzymes still catalyze our thoughts. The cells that pioneered division and cooperation still dance in every heartbeat, every breath. To contemplate the origin of life is to contemplate ourselves, not just our ancestry, but our potential. For life is not static. It is not finished. It is a dynamic, creative unfolding, writing new verses with every generation. And the mystery of its beginning is not a wall but a mirror, reflecting the light of inquiry, of imagination, of being. Beneath the surface of our planet, hidden in the folds of rocks and sediment, lies a story older than any written word. A story etched in, molecules whispered in the language of atoms. This story begins not with grand gestures, but with the humble chemistry of Earth's primordial environment. The early Earth was a world of contrasts, scorching heat and icy cold, violent storms and tranquil pools, chemical abundance and scarcity. From these contradictions arose the first glimmers of life, shaped by processes that challenge the boundaries of our understanding. In the depths of this ancient world, life's ingredients mingled. Simple molecules collided and combined in countless ways, forming more complex compounds. The elements carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, each fundamental to living systems, found pathways to bond and rearrange. The synthesis of amino acids, nucleotides, and lipids set the stage for the emergence of protocells, the precursors to life as we know it. Among the pivotal discoveries in recent decades is the realization that life's origin was not a singular event, but a mosaic of interconnected steps. No single spark created life outright. Instead, multiple chemical networks coalesced, interacting and evolving over time. This holistic view sees metabolism, genetics, and compartmentalization as intertwined threads, weaving the tapestry of life's beginnings. Metabolism, the web of chemical reactions sustaining organisms, likely originated as simple cycles driven by the energy gradients of early Earth. Alkaline hydrothermal vents, with their mineral-rich chimneys and stable conditions, are compelling candidates for life's cradle. These vents provide natural proton gradients and catalytic surfaces that could have powered the first metabolic networks. Here, the boundary between chemistry and biology begins to blur, as molecules acquire the capacity to transform energy and sustain themselves. Parallel to metabolic development, genetic molecules emerged. RNA, with its unique ability to store information and catalyze reactions, stands at the heart of hypotheses explaining life's early evolution. The RNA world hypothesis suggests that before DNA and proteins took center stage, RNA molecules performed both informational and enzymatic roles. Laboratory experiments have demonstrated that RNA can self-replicate under certain conditions, hinting at how life might have taken its first steps toward self-sustaining systems. Compartmentalization followed. The formation of lipid vesicles, self-assembling into membrane-bound spheres, created distinct microenvironments. These protocells could concentrate reactants, protect fragile molecules, and enable more controlled chemical interactions. This compartmentalization was crucial for the transition from chemistry to biology, marking the birth of individuality and selection. As protocells grew and divided, evolutionary forces began to act. Variations in molecular composition, replication, efficiency, and metabolic capability led to natural selection at the molecular and cellular levels. Over countless generations, more complex and efficient systems emerged, paving the way for true cellular life. The story of life's origin is also one of resilience and adaptability. Life thrived despite the Earth's harsh conditions, intense radiation, volcanic eruptions, and fluctuating climates. These challenges shaped the robustness of early life forms and guided their evolutionary trajectories. Looking outward, the principles uncovered in the study of life's beginnings inform our search for life beyond Earth. The universality of chemistry suggests that given similar conditions, life might arise elsewhere. Yet, the diversity of environments that support life on Earth expands our imagination of where it might be found, from the icy moons of Jupiter and Saturn to the subsurface of Mars and distant exoplanets. In contemplating how life began, we glimpse the profound interconnectedness of the cosmos. The atoms in our bodies were forged in ancient stars, scattered by cosmic winds, and gathered by Earth's gravity. The emergence of life is a testament to the universe's capacity for complexity and creativity. This grand narrative invites us to reflect on our place within it. We are both products and participants in the ongoing story of life, a story that began billions of years ago in the quiet chemistry of our planet and continues to unfold in every living moment. Life's beginnings, deeply woven into the fabric of Earth's early history, reveal a saga of chance and necessity, a delicate dance between order and chaos. Among the many theories illuminating this dawn, one concept has captured both scientific imagination and philosophical wonder: the emergence of self-organization. From simple molecules came intricate patterns, not by design but by natural laws guiding their interactions. Self-organization means that, under the right conditions, systems can spontaneously form ordered structures and behaviors. This principle, visible in the formation of snowflakes, the spiral of galaxies, and the flocking of birds, also underpins the origins of life. When molecules reach critical concentrations and interact on catalytic surfaces, new properties emerge, properties that no individual molecule possesses alone. In early Earth's tumultuous environment, mineral surfaces such as clays and iron-sulfur compounds likely played a vital role. These surfaces acted as scaffolds, concentrating molecules and aligning them into configurations favorable for chemical reactions. Experiments show that nucleotides and amino acids can adsorb onto such minerals, enhancing polymerization, the assembly of longer chains essential for life's functions. From these surfaces, the first polymers could form. Polymers like RNA and peptides exhibit the capacity for information storage and catalytic activity. The assembly of polymers was a key step bridging the gap between inert chemistry and the dynamic processes of biology. Another intriguing idea is the metabolism-first hypothesis, which proposes that self-sustaining chemical networks preceded the evolution of genetic information. According to this view, life started as cycles of chemical reactions that harvested energy from the environment, maintaining themselves far from equilibrium. These networks, fueled by Earth's geochemical gradients, generated increasingly complex molecules, eventually giving rise to genetic systems. This contrasts with the genetics-first hypothesis, where replicating molecules like RNA appeared before metabolism was fully established. Modern research suggests these pathways may not be mutually exclusive. Instead, metabolism and genetics could have co-evolved, each reinforcing the other. As molecular complexity grew, so did the emergence of feedback loops. Certain molecules catalyze their own formation or the creation of other molecules, forming autocatalytic sets. Such networks could grow exponentially and adapt to changing conditions, exhibiting a rudimentary form of evolution. The transition to cellular life brought new challenges and innovations. To sustain complex chemistry, early life needed compartmentalization. Membrane-like structures composed of amphiphilic molecules, those with both hydrophobic and hydrophilic parts, spontaneously assembled into vesicles, creating isolated environments. These protocells could maintain different chemical conditions inside and out, allowing for more efficient reactions and protection from harmful agents. Within these protocells, primitive metabolic pathways could operate, using chemical energy, sources like hydrogen sulfide or methane. Some hypothesize that life began near hydrothermal vents, where mineral-rich fluids and temperature gradients provided energy and catalytic surfaces. These vents remain hotspots of microbial diversity. Today, living relics of life's ancient past, the origin of genetic material within protocells was a profound milestone. RNA, capable of both storing information and catalyzing reactions, likely played a central role. Over time, RNA molecules evolved the ability to replicate with higher fidelity, increasing the accuracy of genetic transmission. This laid the foundation for Darwinian evolution, where variation and selection drive adaptation. Eventually, DNA took over as the primary genetic material due to its stability, and proteins became the main catalysts due to their versatility. This division of labor increased efficiency, enabling the development of more complex organisms. The origin of life thus emerges as a story of gradual complexity, where simple molecules organized into systems capable of growth, replication, and evolution. It is a story written not in words, but in chemistry, physics, and time, a testament to the creative power of nature's laws. In seeking to understand this story, humanity continues to explore the boundaries of knowledge. Laboratory experiments simulate early Earth conditions, recreating steps of polymer formation and protocell assembly. Discoveries of extremophiles, living in harsh environments, expand the definition of habitable conditions. Space missions search for signs of life's ingredients on Mars and icy moons. Each new finding deepens our appreciation for the fragile miracle that is life. It reminds us that our existence is part of a vast continuum, shaped by forces both grand and minute, unfolding over billions of years. The tale of life's origin invites us to explore not only Earth's ancient past, but also the cosmic context that made it possible. The atoms composing living organisms were born in the hearts of stars, fused in fiery furnaces and scattered by supernovae across the universe. This cosmic heritage connects us to the very fabric of existence, suggesting that the seeds of life are woven into the cosmos itself. Within this vast expanse, the conditions for life's emergence are rare, yet not unique. Earth's position in the habitable zone, where liquid water can exist, alongside its protective atmosphere and magnetic field, created a cradle for life. Water, the solvent of life, provided a medium where molecules could move, collide, and react with remarkable freedom. The presence of organic molecules in interstellar clouds and meteorites hints that the building blocks of life were delivered to Earth from space, enriching the primordial soup. This exogenous delivery may have complemented Earth's own chemical processes, accelerating the path toward life. As research continues, new frontiers emerge. The field of astrobiology seeks to understand how life might arise elsewhere, examining environments that challenge terrestrial assumptions. The discovery of extremophiles, organisms thriving in boiling acid pools deep underground or under intense radiation, broadens the horizons of habitability. These findings inspire us to look beyond Earth, to moons like Europa and Enceladus, where subsurface oceans may harbor life beneath icy crusts. The search for biosignatures, chemical or physical signs of life in the atmospheres of exoplanets grows ever more sophisticated. On a microscopic scale, the study of synthetic biology and origin of life chemistry continues to push boundaries. Scientists attempt to recreate lifelike systems in laboratories, testing hypotheses and unraveling the delicate balance of factors necessary for life. These experiments shed light on the minimal requirements for life and help to define the transition from chemistry to biology. Understanding life's origin also carries profound philosophical implications. It challenges us to reconsider what it means to be alive, to see ourselves as part of a continuum rather than isolated beings. Life's emergence from the inanimate blurs the lines between non-life and life, revealing a spectrum of complexity. In this light, life appears as an inevitable consequence of the universe's physical laws, an expression of self-organization and emergence. Yet it is also a unique and precious phenomenon, shaped by contingent events and environmental constraints. Our curiosity about life's beginnings is more than scientific inquiry. It is a quest for connection. Through understanding where we come from, we gain insight into our place in the cosmos and the responsibilities that come with consciousness. The story of life's origin is a beacon, illuminating the path from chaos to order, from simplicity to complexity, from chance to purpose. It reminds us that within the vast universe, life has found a way to awaken, to flourish, and to ponder its own existence. The narrative of life's emergence also reveals the profound role of energy flow and thermodynamics. Life is not merely a collection of molecules, but a system sustained by a constant exchange of energy, maintaining order amid the natural tendency toward disorder. This balance, described by the second law of thermodynamics, is central to understanding how life can arise and persist. Early Earth presented abundant energy sources: sunlight, volcanic heat, chemical gradients, and electrical discharges. These energy inputs drove reactions far from equilibrium, creating conditions ripe for complexity. Chemical systems harnessed these flows, organizing into networks that could capture and utilize energy efficiently. In particular, the concept of dissipative structures illuminates how order can arise spontaneously in open systems exchanging energy with their environment. Such structures maintain and increase their complexity by dissipating energy gradients, exemplified by hurricanes, convection cells, and living organisms. The earliest proto-metabolic networks likely operated as dissipative systems, using environmental energy to drive synthesis and replication. This dynamic allowed them to grow, adapt, and evolve. Energy flow became the engine of life, fueling the continuous renewal that characterizes living systems. One of the mysteries is how this flow became coupled to information storage and reproduction. The interplay between metabolism and genetic material gave rise to systems capable of heredity and evolution. RNA molecules with catalytic and informational roles embodied this dual function, bridging chemistry and biology. Understanding energy's role in life's origins also informs the search for extraterrestrial life, environments rich in chemical energy, such as hydrothermal vents, may harbor life independent of sunlight. This expands the possibilities beyond the traditional habitable zone. The delicate balance of energy capture and dissipation is mirrored in contemporary biology, where cells maintain order by consuming nutrients and expelling waste. This principle links the ancient origins of life to the complex organisms of today. Moreover, the study of prebiotic energy transduction informs synthetic biology and the design of artificial life. Replicating these processes in the lab deepens our grasp of life's essence and the conditions necessary for its emergence. Through the lens of thermodynamics and energy flow, life emerges as a natural phenomenon, an exquisite dance of matter and energy shaped by universal laws. It is a reminder that the spark of life is more than chemistry. It is a process woven into the cosmos's unfolding fabric. Within the vast panorama of hypotheses about life's origin, the lipid world hypothesis offers a captivating glimpse into the crucial role of membranes. Lipids, with their unique chemical structures, naturally form bilayers that enclose and protect chemical systems, creating distinct internal environments. This boundary, a membrane, is fundamental to life. It defines the individual, allowing controlled exchange with the surroundings. Before genetic materials dominated, early Earth may have seen the spontaneous formation of lipid vesicles. These microscopic bubbles acted as containers for biochemical reactions, enabling primitive metabolism and molecular interactions to occur more efficiently. Such compartments concentrated molecules, increased reaction rates, and shielded fragile polymers. From the chaotic external environment, the formation of protocells, membrane-bound precursors to living cells, represented a milestone. These protocells could grow, divide, and inherit molecular components, fulfilling the criteria of life in a rudimentary form. Membrane dynamics, including permeability and fluidity, influence the ability of protocells to interact with nutrients and energy sources. Research shows that lipid membranes may have facilitated the selection of functional molecules. Membranes could capture and retain RNA or peptides, enhancing their catalytic capabilities and enabling early evolutionary processes. This interaction between compartmentalization and molecular function laid groundwork for increasing complexity. Additionally, mineral surfaces likely interacted with these lipid structures, providing catalytic sites and structural support. The synergy between lipids, minerals, and organic molecules formed a fertile environment for life's genesis. The lipid world hypothesis complements other models by highlighting how physical structure enabled biological function. Without membranes, the delicate chemistry of life would dissipate, unable to sustain the complexity necessary for evolution. In modern biology, membranes continue to be central, regulating cellular processes, communication, and energy transduction. Understanding their origin offers insight into the fundamental architecture of life and the transitions from chemistry to biology. This perspective emphasizes that life is more than genetic information. It is an integrated system where structure and function co-evolve, where boundaries define identity and enable interaction. Exploring membranes' origin deepens our appreciation of life's intricate design and the subtle processes that transformed Earth's chemistry into the vibrant biosphere we inhabit. As the story of life's beginnings unfolds, the question of heredity emerges as a cornerstone. How did early life store, transmit, and evolve information? Among the prevailing scientific narratives, the RNA world hypothesis offers a compelling framework, positing that ribonucleic acid molecules preceded DNA and proteins as both genetic carriers and catalysts. RNA is remarkable for its dual functionality. It can store genetic information like DNA and catalyze chemical reactions like proteins. This versatility suggests that early RNA molecules could replicate themselves, catalyze reactions necessary for their synthesis, and guide the assembly of other molecules, setting the stage for biological complexity. Laboratory experiments have demonstrated that certain RNA sequences, called ribozymes, can catalyze their own replication and other chemical transformations. This discovery provides experimental support for RNA's central role in early life, bridging the gap between chemistry and biology. In the primordial environment, pools of RNA-like molecules may have formed through spontaneous polymerization, nucleotides. These molecules would undergo selection, where variants with improved replication or catalytic efficiency outcompeted others, initiating Darwinian evolution. Over time, the RNA world gave rise to more stable DNA molecules for information storage and versatile proteins for catalysis. DNA's superior stability allowed for longer, more reliable genetic codes, while proteins expanded the range of biochemical functions. The transition from RNA to DNA protein life was gradual, involving complex interactions and innovations. This shift marked a fundamental evolution in life's machinery, enabling the diversity and complexity seen today. Understanding RNA's role illuminates the intimate relationship between genetics and metabolism, revealing how life's earliest systems integrated information and function. It also guides current research in synthetic biology, where scientists design RNA-based systems to emulate and explore life's origins. The RNA world hypothesis, while powerful, is part of a broader mosaic. It intersects with theories about metabolism, membranes, and energy flow, emphasizing life's multifaceted emergence. In contemplating RNA's legacy, we see how life harnessed molecules to not only exist but to remember, reproduce, and adapt, a remarkable leap from mere chemistry to biology. The emergence of replication, heredity, and metabolism inevitably leads to the phenomenon we recognize as evolution, a process central to life's unfolding complexity. Early replicators, be they RNA molecules or protocells, existed in a dynamic environment shaped by competition and cooperation, mutation and selection. Evolution operates as a natural filter, favoring entities that are more efficient at surviving and reproducing. Mutations introduce variation, some beneficial, some detrimental, but through countless generations, advantageous traits accumulate. This iterative process fosters the gradual emergence of complexity, diversity, and adaptation. In the prebiotic world, evolution might have begun with simple molecular replicators, progressing to protocells capable of metabolic functions and reproduction. The formation of genetic codes enabled more accurate inheritance, setting the stage for complex organisms. Selection pressures shaped early life forms, driving innovations such as improved replication fidelity, energy utilization, and environmental resilience. Cooperation between molecules and protocells likely enhanced survival, leading to symbiotic relationships that became the foundation for cellular life. The concept of major transitions in evolution highlights pivotal steps where simpler units combine to form more complex wholes: molecules to protocells, protocells to cells, cells to multicellular organisms. Each transition brought new levels of organization and novel capabilities. This evolutionary framework explains not only life's origin, but also its ongoing transformation. From microscopic beginnings, life has diversified into the astonishing variety of forms populating Earth. Understanding evolution's roots offers insight into life's resilience and adaptability. It reveals how life is not static, but a continuous journey shaped by environment, chance, and necessity. This process, written into the very molecules and cells that compose us, connects all living beings in a shared story of change and survival. Evolution is the engine that propelled life from humble origins to the vast biosphere we witness today. Amid the vast expanse of Earth's history, the rise of cellular life marks a defining chapter. Cells, the fundamental units of life, encapsulate the chemistry of life within membranes, organizing biochemical reactions, storing genetic information, and sustaining metabolic processes. Two primary domains of life, bacteria and archaea, represent some of the earliest branches in the tree of life. These single-celled organisms exhibit remarkable diversity and adaptability, thriving in environments from boiling vents to frozen deserts. The origin of cells likely involved the encapsulation of self-replicating molecules within lipid membranes, forming protocells that gradually gained complexity. Membranes not only provided physical boundaries, but also created controlled environments essential for metabolism and genetic functions. As cells evolved, they developed sophisticated mechanisms for energy production, including photosynthesis and respiration. Photosynthetic bacteria, harnessing sunlight to convert carbon dioxide and water into organic molecules, transformed Earth's atmosphere, paving the way for oxygen-dependent life forms. The emergence of oxygenic photosynthesis triggered the Great Oxygenation Event, radically altering Earth's environment. This shift enabled aerobic respiration, a more efficient energy-producing process, fostering the evolution of complex multicellular life. The transition from simple prokaryotic cells to eukaryotes, cells with nuclei and organelles, remains a pivotal evolutionary milestone. The endosymbiotic theory explains this leap, proposing that ancient prokaryotes engulfed others, forming symbiotic relationships that evolved into mitochondria and chloroplasts. This cellular complexity allowed for larger, more specialized organisms, setting the stage for the biodiversity we observe. The interplay between genetic innovation, energy capture, and environmental change drove life's trajectory. Understanding cellular origins bridges chemistry and biology, illustrating how life's defining characteristics coalesced. Cells are living laboratories, dynamic systems maintaining order and adapting through molecular networks. This chapter of life's origin invites awe for the intricate processes that transformed inert matter into vibrant, self-sustaining entities capable of growth, reproduction, and evolution. The genesis of multicellularity stands as another profound turning point in life's story. From solitary cells emerged complex organisms composed of countless cells working in harmony, a remarkable leap in biological organization. This transition allowed for specialization, division of labor, and increased size, features essential to the rich diversity of life forms. Multicellularity likely evolved multiple times independently, reflecting its adaptive advantage. Initially, cells aggregated for mutual benefit, protection, resource acquisition, or environmental resilience. Over time, these associations became more integrated, with cells differentiating into distinct types performing specialized functions. This cooperation required intricate communication mechanisms, ensuring cells' coordinated activities for the organism's survival and reproduction. The evolution of signaling pathways, gene regulation, and cellular adhesion underpinned this integration. With multicellularity came new challenges, including maintaining homeostasis, managing waste, and reproducing as a collective. Innovations, such as tissues, organs, and developmental programs arose to meet these demands, enabling organisms to exploit diverse environments. The fossil record offers glimpses of early multicellular life, such as the Ediacaran biota, whose enigmatic forms hint at life's experimentation with complexity. These early organisms laid foundations for the explosion of life forms in the Cambrian period, when body plans diversified dramatically. Multicellularity exemplifies life's capacity to transcend simplicity, forming systems where the whole is greater than the sum of its parts. It embodies cooperation amid competition, fostering resilience and adaptability. This evolutionary milestone reflects the deep interconnectedness of life, where individuality merges with community, and cellular selves unite to create new identities. In contemplating multicellularity, we witness life's enduring drive toward complexity, diversity, and innovation, an ongoing narrative that continues to shape our existence. As the saga of life's origins approaches the present, it reveals a continuous, unfolding process shaped by countless factors, both terrestrial and cosmic. The Earth's early environment, rich in chemical ingredients and energized by diverse forces, provided the stage for life's inception. From the spontaneous assembly of molecules to the emergence of complex evolving organisms, the journey reflects a tapestry woven by chance, necessity, and natural laws. Modern science continues to unravel this intricate story, using interdisciplinary approaches that blend chemistry, biology, geology, and astronomy. Each discovery, be it a novel molecule in a meteorite, a new fossil species, or insights from molecular biology, adds depth and nuance to our understanding. The origin of life remains a profound mystery, inviting wonder and humility. It challenges us to consider not only how life began, but what it means to be alive, to persist, to evolve. This quest transcends scientific inquiry. It touches on philosophy, spirituality, and the human desire to connect with the cosmos. It reminds us that we are part of a vast interconnected web, a continuation of ancient chemical dances that began billions of years ago. As we gaze at the stars, search for life beyond Earth, and explore our own biology, we participate in this grand story. The origins of life are not just a chapter in the past, but a living, evolving narrative, inspiring curiosity and awe. In understanding where we come from, we gain insight into the fragile beauty and remarkable resilience of life itself. It is a story without end, a profound testament to the power of nature and the mystery of existence.